1, 2. It's okay. So, hello everybody, my name is Mathias Witte Paz, and today I'm here to present Evidente. So, when we face a project where we would like to analyze a group of strains coming from one organism, one of the first steps could be to reconstruct a phylogenetic relationship among these strains. And this can be done by using the single nucleotide polymorphisms. So, in order to do this, we first align all of the genomes we have against a common reference, and then we use all of the columns that have at least one mutation in order to reconstruct such a phylogenetic tree. This tree could look a little bit like this. This is the phylogenetic tree of the Mycobacterium leprae from the study from Schoenemann et al. In 2018. But what about afterwards? I mean, what if we're interested in knowing the relationship among the Snips and the reconstructed phylogenetic tree? Or if we're interested in just one specific subtree of the tree, and we would like to know if there's some enriched characteristics within this phylogenetic tree that might not be represented among the whole phylogenetic tree. Or a third question, if we're interested in two subtrees, and we would like to know if there are common SNPs between these two subtrees, these questions are not easy to answer at this point. In order to answer these questions, we develop Evidente. Evidente stands for an efficient visual analytics tool for data enrichment in SNP-based phylogenetic trees. Evidente was mainly developed in JavaScript and uses also phylocanvas for the visualization. To answer the questions I've already mentioned, Evidente provides a user an interactive visualization of the phylogenetic tree and the SNPs the reconstruction was based on, and furthermore allows the user to run an enrichment analysis on the subtrees of the phylogenetic tree. One of the first definitions I would like to clarify is the clade specificity of a SNP. So first, a SNP is defined as such a column that we're using from our alignment, and if this SNP appears on the same form among all of the members of one specific taxa, then we call that clade specific. As we can see here, this SNP1 is appearing on the same form among the whole members of the left clade, while and also in the same form on this clade. While on the other hand, the SNP2 is distributed among the whole phylogenetic tree. For this reason, we call the left SNP a supporting or a clade-specific SNP, and a non-supporting SNP to the SNP2, since it's not supporting the decision to use or to use this reconstructed phylogenetic tree to represent our data. So this is an important definition, since from the from a pre-computed Newick file and from a pre-computed SNP table, we're able to first compute the clade specificity of the SNPs we're analyzing, and these 3 points would make the main input of Evidente. If the user has further metadata that he would also like to include in his analysis, he can also upload this information to Evidente so he has the whole information within one interface. At the end, what we would like to visualize is the phylogenetic tree the SNPs reconstruction was based on, and somehow encode the clade specificity of the SNPs. And additionally, we'll also like to visualize the distribution of possible enriched features among the phylogenetic tree, such as Go terms or genes that might be affected by our SNPs. So to visualize these elements first, for the phylogenetic tree, we choose the common approach of a dendrogram, and we also use the interactive node for phylocanvas to encode more information that the user had provided. For the SNPs, we visualize them as rectangles aligned to the corresponding taxa, and we're coloring them in order to encode the clade specificity and also the base they're mutated to. And lastly, for the distribution among the whole phylogenetic tree, we're highlighting the least common ancestors of those taxa that have this enriched characteristic, so the user can also visualize this. So this returns the main interface of Evidente, which is this one. In the main canvas, we can visualize the phylogenetic tree, and on the right side, we have the further tools or information that Evidente is going to provide the user. One of the first features I would like to discuss is the visualization of the SNPs. As we can see here, we are visualizing two supporting SNPs since they are colored and also appearing only within one clade. On the other hand, we're also visualizing two non-supporting SNPs since they're distributed among the whole phylogenetic tree, as we can see here. From the interactive nodes, we are also able to get more information on the splits we're interacting with that might support this split we're visualizing. And finally, we also have lists that are going to classify the SNPs that are within this phylogenetic tree depending on their clade specificity, and we're also able to compute a list of those common SNPs between two subtrees as the ones shown here. Second feature I would like to present is the filtering of the taxa. So if the user was able to upload further metadata of the taxa, he can also use the criteria to focus only on those taxa he's interested in. So as we can see here, Evidente is able to collapse all of the taxa that do not fulfill the criteria so that the user can focus only on those taxa he's interested in. And finally, I would like to present the enrichment analysis. So as I've already mentioned, if we're interested in knowing if within one specific subtree of our tree we have enriched characteristics, as this case genes that are affected by our SNPs, Evidente allows the user to run an enrichment analysis based on the Fisher's exact test, and it's going to return such a list from all the features, all the characteristics with a corresponding p-value. From this list, the user is also able to visualize the distribution among the whole phylogenetic tree, as we can see here, by highlighting the different nodes that contain this enriched characteristic, and also is able to visualize the SNPs that leads to this enrichment. Evidente is still an ongoing work, and there are some points we will also like to improve. And one of the first is the visualization. Even though Evidente is able to interact with large phylogenetic trees, the resulting visualization of it is not as good as it should be. We will also like to make Evidente more adaptable for the interface, because for some window sizes it's not working correctly. Finally, we will also maybe incorporate interactive parsing methods to be less restrictive for our analysis, or maybe include the analysis for further characteristics such as gene pathways. And the last point would be to implement further enrichment analysis methods and not just only provide the Fisher's exact test, so that the user can have a further or more options for his analysis. This would be the point, all the features that Evidente provides. And if you would like to test the tool, here's the link for its availability. Or if you have more questions or would like to discuss more on the features of Evidente, you can also come by our poster. Thank you very much.
A very nice talk.
Thank you.
Can I ask about the Fisher exact test?
Yeah.
Is it realistic to, so those are interactive selections that you make and then look at enrichment within them?
So you choose first, you choose first a subtree that contains the node. So we are choosing just all the specific, so we're classifying it within this subtree or not within the subtree, and having this having this characteristic or not having this characteristic. So that's the form of our contingency table.
So it's an in-out control.
Yeah.
Where you specify the in as the subtree, and then the rest of the tree is the out.
Exactly.
Okay, that's important.
Yeah.
Thank you for clarifying that.
That's a nice tool, and it's primarily going to be useful for bacteria that have like a single chromosome or none.
Yeah.
Like
at this point, we're only testing prokaryotes. I don't think it's going to be.
But in the case of like something like mycobacteria, you're going to at some point get selection occurring in your isolates from drug resistance. So is there a way to include, like in the filtering step, known targets of drug resistance and filter those out?
How would we encode this characteristic?
Well, so there are given genes that we know with specific mutations that, I don't know how you would encode it. I'm just saying that like these homoplastic mutations will arise in the data set.
We haven't thought about it, but it will also be able, I think there's not that complicated to implement or to encode with it or to add for the analysis. So that would be actually a good feature to include.